Bismillahirrahmanirrahim. This is me, Muhammad Asif, and I am working as a lecturer in the Institute of Paramedical Sciences, Hyder Medical University. Dear viewers, today I came up with another important topic related to immune hematology. But before explaining the topic, we can see in the picture that there is a soldier, and the duty of the soldier is to protect the country. So the topic is also related to the same picture, and there are certain substances in our body which protect our body from pathogens, and these substances, and one of these substances is immunoglobulin. As the name indicates, that immunoglobulin is the combination of two words, immuno and globulins. Immuno is derived from immune system, and globulins are proteins. So we can say that immunoglobulins are the proteins of immune system. Immunoglobulins are also known as antibodies. So antibodies or immunoglobulins are those substances which is produced by a body against specific stimulus. This is a protein electrophoresis of serum. So this graph shows that in the plasma or serum there is highest concentration of albumin, and there is little percentage of globulins. There are various types of globulins, for example alpha 1, alpha 2, beta and gamma. Immunoglobulins belongs to the gamma globulins, which are present in the plasma or serum. As we discussed, that antibodies are substances that are produced against specific stimulus, and that stimulus is known as antigens. So what is antigen? So antigens are those substances which stimulate the immune system, and the immune system then produce antibodies. Now there are two types of antigen. One is self-antigen and the other is non-self-antigen. Self-antigen are those antigens which are recognized by our immune system. For example, ABO blood group antigens or RH blood group antigens are all types of blood antigens. They are present on the surface of red blood cells, but our body do not produce antibodies against those antigens. Why? Because our body knows that these are our self-antigens. That's why antibodies are not produced against those antigens. While non-self-antigens are those antigens which are not recognized by the immune system. So if our immune system do not recognize those antigens, so antibodies will be produced. But a question arises that why in autoimmune hemolytic anemias or autoimmune thrombocytopenia, why antibodies are produced against self-antigen, although these are the antigens of our own cells. So why? So the reason is that sometime a small molecular weight substance attached to the surface of an antigen which change the structure. So when the structure is changed, so our body then recognize those antigen as non-self antigen. That's why antibodies are produced. Otherwise antibodies are not produced against self antigen. Bacteria is a non-self antigen, so antibodies are produced against them. Viruses and etc. etc. Difference between immunogen and antigen. An immunogen is a substance that must stimulate the immune system, while antigen may or may not stimulate the immune system. So we can say that all immunogens are antigens, but all antigens are not immunogens. Epitopes and haptens, there is a little bit of difference between them. Epitopes are basically a portion of an antigens that are presented to antigen presenting cell, that is macrophage, while haptens are low molecular weight substances that cannot itself stimulate the immune system, but when it combines with another molecular weight substance and its molecular weight increases, then it can stimulate the immune system. Now there are certain factors that affect the antigenicity of an antigen, and these factors are size of the antigen, nature of the antigen, dose of the antigen, route of entry of the antigen, and degradability. So size of the antigen is important because those antigens whose molecular weight is very small, that cannot be recognized by the immune system, and antibodies will not be produced against that specific antigen. But if an antigen is too large, again the immune system cannot recognize it. So those antigens whose molecular weight is between 8,000 to 10,000 Dalton, that antigens are best antigens, and the immune system will produce antibodies against them. The other thing is nature of antigen. Nature of antigen means that some antigens may be protein, some antigen may be glycoprotein, some antigens may be polysaccharides or carbohydrates, some antigens may be glycoprotein. So those antigens that are protein in nature, antibody will be efficiently produced by the immune system. Second, those antigens whose nature is glycoprotein, antibodies will produce again them efficiently, and so on. Number third is dose of antigen. If antigen is introduced into the body in small concentration, so lesser amount of antibodies will be produced as lesser antigens will be introduced into the immune system. For example, vaccination. In vaccination, for example, on day first, a person is injected with vaccine. After 1 month, he is again vaccinated, and after 3rd month, he is again vaccinated. Why? In the first dose, sometime the antigen are in small concentration, in the third antigen are in highest concentration. So when there is smaller amount of antigen, so the immune system will produce antibody in smaller concentration. And the other thing is route of antigen. Route of antigen means that from where the antigen will be entered into the body. For example, through mouth, intravenous or intravascular. So those antigens that are introduced are injected through subcutaneously, they will reach to the immune system rapidly as compared to intravascularly or through mouth. Because when you introduce an antigen subcutaneously, they will enter through the lymphatic system, through into the lymph nodes where it is presented to the T cells. Number 5, those antigens that are easily degradable and easily phagocyted by the phagocytic cells, they will easily present it to the immune system and antibodies will produce against that antigen. Now, structure of antibodies. As we discussed that the function of antibody is to protect the body against foreign substances or antigens. Now this is the structure of an antibody. We can see that this is a Y-shaped structure. This is a Y-shaped structure which is composed of 4 chains, 1, 2, 3 and 4. These are 4 chains. As we discussed earlier that immunoglobulins are glycoproteins which is composed of proteins and carbohydrate. So this structure, this chain is composed of amino acid and these are oligosaccharides. So this antibody is basically a glycoprotein. This structure is composed of 2 identical chains. 2 smaller, this one, blue and light blue color, and this one, the orange color. This chain is longer than this one, so therefore this chain is known as heavy chain. And this one is smaller, that's why this is known as light chain. Heavy chain is called heavy chain because the molecular weight of this heavy chain is higher than this one. And this chain consists of 420 amino acid. And the molecular weight of this chain is from 55,000 to 77,000 Dalton. That's why this chain is known as heavy chain. The light chain consists of 212 amino acid and the molecular weight of this chain is 25,000 Dalton. So therefore it is known as light chain. Both these heavy and light chain are further divided into segments or fragments. Like this one, this is dark blue, this is light blue. So dark blue is known as constant region of light chain, CL, while the light blue is variable region of light chain. This light blue is further divided into some segments like this white. This is the hypervariable region of the amino acid. Although there is variation in the amino acid in the variable region, but this portion and this portion consists of 110 to 120 amino acid, which is known as hypervariable region. Similarly, in the heavy chain there is also hypervariable region. Now the constant region is further subdivided into segments. For example, this is again variable region, this is constant region 1, these are constant region 2, this is constant region 3. This portion is known as Fab region. Fab antigen binding site. These are known as basically antigen binding site. If AB stands for antigen binding fragments. So antibodies basically attach through these portions to the antigen, and this portion is known as constant region, FC region. Fragment of crystalline, this is known as FC region, and this region is responsible to attach to the surface of a cell. For example, this portion have receptor on various cell. Let's suppose placenta. If we consider this antibodies as IgG antibodies, which we will explain later. So IgG antibodies will have specific receptor on the surface of placenta. Similarly, on the surface of macrophage, there are specific receptors present on the surface of macrophage for this antibody. Now, these four chains are connected to each other, so antibody should be stable and will be present in a compact form. So therefore, this is disulfide bond. Disulfide bonds are present in between this chain and also there. This portion is known as hinge region of the antibody, or we can say that neck region of the antibodies. And antibodies may become flexible depend upon the type of antigen. So this is the overall structure of an antibody. Now, types of antibodies. Antibodies are divided into 5 major types, and these are divided on the basis of heavy chain. There are 5 different types of heavy chain. Gamma, mu, alpha, delta and epsilon. So based on these heavy chains, we divide immunoglobulin into 5 types. Gamma heavy chain, so immunoglobulin G. Mu heavy chain, so immunoglobulin M. Alpha heavy chain, so immunoglobulin A. If antibody contains delta chain, so immunoglobulin D. If an antibody contains epsilon chain, so immunoglobulin E. There are 5 types. Immunoglobulin G. The structure of immunoglobulins is monomeric, which is composed of single chain, this one like Y shape. There are certain properties of IgG antibodies. For example, IgG is the most abundant antibodies, which consist of 85% of the immunoglobulin, and it is monomeric. IgG antibodies have one most important property, and that property is that it can cross placenta. Why? Because number 1, immunoglobulin G is monomeric, having low molecular weight, and the other reason is that this IgG have specific receptor on surface of placenta. That's why immunoglobulin G can transfer through placenta. And immunoglobulin G can also fix complement system. It means that it can activate the complement system. The other important property is that this antibody is also known as opsonin antibodies. What is opsonin antibodies? Opsonin antibodies are opsonin substances. When get attached on the surface of an antigen, it helps the phagocytic cells to engulf it. So those antibodies or those substances are known as opsonin substances, and that process is known as opsonization. That is the attachment of the antibody on the surface of antigen. In the whole process, when it engulfs it, that process by the phagocytic cell, that process is known as phagocytosis. Another property that IgG antibodies are incomplete antibodies. Incomplete antibodies are basically those antibodies which cannot form visible agglutination reaction at saline phase. So that antibodies are also known as IgG antibodies or incomplete antibodies. So IgG antibodies is incomplete antibodies. And IgG antibodies are also known as warm antibodies. Why? Because it gives best reaction at 37 degrees centigrade. So this antibody is known as warm antibodies, which react best at 37 degrees centigrade. There are 4 subclasses of IgG antibodies: Ig1, Ig2, Ig3 and Ig4. Ig3 antibodies can efficiently activate the immune system and activate the complement system, followed by Ig1, then Ig2, and Ig4 may and may not activate the complement system, but most literature said that IgG antibody cannot activate the complement system. IgM is one of the most important antibody, which is known as IgM based on mu chain. So we can say that there are 5 monomers: 1, 2, 3, 4 and 5. So as these have 4 monomers, so we call it as pentameric antibody. These 4 monomers are attached, are joined together by another chain. This chain is known as J chain. IgG do not have J chain. These antibodies can attach to 10 antigens at a time. One antigen through this, 2, 3, 4, 5, 6, 7, 8, 9, 10. So 10 antigens can be combined with IgM antibodies at a time. Properties of IgM antibodies. IgM antibodies is the third antibodies that are present in highest concentration in serum. IgM antibodies are also known as primary response antibodies. When an antigen enters first time into the bodies, so the first antibody will produce, and that antibodies will be IgM antibodies. IgM antibodies can efficiently activate the complement system, but these antibodies cannot cross placenta because this antibody is pentameric antibodies having high molecular weight, having high structure. Further, these antibodies having no receptor on the surface of placenta, and these antibodies are complete antibodies. Although IgM antibodies can give reaction below 37 degrees centigrade, but it gives best agglutination reaction at 4 degrees centigrade. Therefore, this antibodies is known as cold antibodies. And this antibody is also known as agglutinin antibodies because these antibodies give visible agglutination reaction at saline medium, which is known as complete antibodies. IgA antibodies are mostly found in secretions like saliva, like milk, body fluids. So therefore this is also known as secretory antibodies. These antibodies can be present in monomeric form or dimeric form, but in the serum 90% of IgA antibodies are present in monomeric form, while 10% in dimeric form. In the dimeric form, there is again 2 monomers are combined through a chain that is known as J chain. These are the properties of IgA. That it is second highest antibodies present in the serum. It does not fix complement system, cannot cross placenta, and react at 37 degrees centigrade. Just like IgG, As IgG have 4 subtypes, similarly IgA also have 2 types, IgA1 and IgA2. IgD. IgD is a monomeric antibody, and this is the 4th highest serum IgG antibodies. IgD antibodies are present on the surface of B cells, and this antibody cannot activate the complement system. IgE antibodies is the most important antibody, especially found in allergic conditions. So structure wise, it is a monomeric antibody, and one property is that it have an extra domain constant region 4. This is the least antibody present in the serum, mainly found in allergic condition, and these antibodies do not fix complement system. Thank you so much.